Okay, hi everyone. Let's go ahead and start our lecture. So for the remaining weeks of our course, I think this is, like I said before, as we get towards the end of the course, we get to talk about things that I think many of the students enjoy learning about more. Um, and this is because, through the course of the semester, we've learned all about different types of mechanisms of how different proteins work together in the immune system, as well as how different cells, our immune cells, cooperate. And laying down all that kind of complicated groundwork has been all the hard work that you guys have been doing, learning about immunology these past, 12 weeks or so. So now we get to put some of this together in ways that really reveal why the stuff that we are learning about is so important in terms of relating to our everyday lives. I think one of the best ways to learn about things is to appreciate how everyday things that you are used to are really tied in with what you're learning about in the class. And so one of these areas has to do with allergies. I think all of us are familiar with allergies. Many of you probably even have certain allergies to things that you've lived with during your life. And often maybe you've wondered, what is the role of your immune system in your allergy? Why do allergies happen, right? And what's the cause of allergies? So that's what we'll talk about in the next couple of lectures. Just as a quick reminder, I think I mentioned to you last time, I have this new computer set up on my desk in my office, and I'm still trying to figure out how to get Zoom to work well with it. So at any point during the lecture, if it seems like the slides are not progressing properly, just shoot me a message in the chat box so I can be aware of that, and hopefully I can fix it. All right. But so far, it seems like it's working okay. So allergies, or the types of reactions that we consider to be allergic reactions, are termed hypersensitivities. Right? So in other words, an oversensitivity to something. One thing that I've asked you to think about this semester is that if our immune system is designed to be able to detect things that don't belong in our body, foreign antigens, for example, as a way of being able to quickly determine that maybe like a pathogen has infected our tissues, how is it that different types of antigens that we're exposed to every day that get into our body don't actually cause us to have full-blown immune reactions? For example, foods. Most foods that we eat, right, are loaded with different types of antigens that certainly would be different than antigens that are coming from inside of our own body's tissues, and things in the environment, things that we breathe in, right, as we go about our daily lives. These types of things, right, are also very different than maybe antigens that your immune cells have built up a self-tolerance against, that your immune cells have been trained to recognize as being okay, right? So why is it then that most foods and environmental antigens don't trigger an immune response? The key, again, has to do with inflammation. Right? I think throughout the semester, you've come to appreciate that inflammation serves as a very important signal to the body that something really is infecting, right? Because an infection with a pathogen causes inflammation in the form of cells being damaged or compromised, and those damaged cells will release certain types of cytokine signals like interleukins that cause inflammation to happen. And that is the sign, right, that something really is an infection and not just some random thing that doesn't really matter that got into your body but might just harmlessly pass its way out. Right? So the key here is that tissues typically avoid inflammation against benign molecules, right? If it's just some sort of foreign molecule that doesn't actually cause any harm, that is not associated with any kind of damage, then there's no reason to believe there would necessarily be inflammation. However, there are certainly times when harmless molecules do trigger a response, right? And that is what hypersensitivity and allergies are all about. So in these particular cases, if you do have a response against some sort of otherwise benign, harmless molecule, you might have a sensation of illness when no infection really exists. And you know what I'm talking about, right? Sneezing, rashes, itchy eyes, coughing, that type of stuff, right? Or even worse, right? We'll talk about some of the symptoms that are associated with allergic responses and understand why some of them actually fit very well with our understanding of what our immune system is mistakenly trying to do. Right? These types of allergic reactions can be just sort of mild and annoying, right? Or they can be deathly serious. So here's a question. When you look at these illustrations below, these are some common sort of everyday examples of allergies. For example, we might be allergic to inhaled materials, such as plant pollen, right? Or dust mites, right? So if you're allergic to dust, and how like underneath beds or sofas, there'd be a lot of buildup of dust, and people might be really sensitive to that. It's not necessarily so much the dust itself, but it's the organisms that live in the dust, like dust mites, right? And then the droppings of these dust mites, as sort of gross as this may sound, is what ends up getting breathed in when you click up a cloud of dust, right? So oftentimes it's the dust mite feces that serve as a foreign antigen. Right? Ingested materials, right? Of course, we think about things like food allergies, allergies to peanuts or other types of tree nuts, allergies to shellfish, whether shrimp, crab, etc. Injected materials. So when I say injected materials, they can be things that are injected into our skin from something like a insect bite or a insect sting, right? Or it might be injected in the form of certain types of drugs, right? And now I say drugs, I don't necessarily mean, illicit or illegal recreational drugs. I'm talking about things like medications, right? Plain old everyday medications that can be injected in. They may cause allergic responses. And also materials that you just contact. So for example, the oils from certain types of plants like poison ivy, the type of rash that forms from that is very much an allergic reaction. Okay? Or certain types of metals like nickel can actually cause an allergic reaction. So in all four of these types of antigen presentation to our immune system, right, is the hypersensitive response that our immune system generates, right? Is this more a product of our innate system or our adaptive immune response that's making some sort of a mistaken judgment call, right? That somehow your body is under attack by these things, when in reality, these antigens are not posing some sort of infectious threat. So is it our innate system or our adaptive system that's responsible for allergies? So it turns out allergies are due to adaptive immune responses, right? It's our adaptive side of our immune system that's making mistakes. Right? So your system makes a mistake in seeing something that's benign and treating it as though it's actually a threat. Right? So therefore, harmless antigens in the air, in the food, or things that we touch are treated as though they are pathogenic. And these types of antigens, then, we just broadly call them allergens, right? These are the things that trigger an allergic response. So consider different types of scenarios here, right? Whether you're talking about allergies to pollen, allergies to pets, right? Allergies to things like different types of foods. Right? These are all examples where there really is no threat, but your adaptive immune system mistakenly believes that there is and responds accordingly. So people who are very sensitive and have a predisposition to developing allergies are called atopic individuals. Now, this is hardly rare at all. In fact, in developed countries like the US, about 40% of the general population can be considered to have some degree of atopic sort of sensitivities to things. I mean, you know this, right? People all around you, friends, family, maybe even yourself, have certain allergies, right? And it seems like there's a tendency to develop certain allergies easier. But the important key here is this is 40% of people in developed countries, right? Children in these countries, so we're talking about first world countries like the United States or places like Western Europe, right? Children in these countries have more allergies than the previous generation before them. So it seems like over the past few decades, with each generation of kids, we're seeing more and more of these allergies. And so what is the cause of this sort of epidemic of allergies? Right? That's one of the more fascinating things that we'll kind of explore between this lecture and the next one. So I wanted to mention this because allergies, like I said, right, it's something that we just sort of accept as part of our daily lives. You know, so these scenarios up here, I think you can easily relate to, right? Although I do have to say, right, I'm very sympathetic to the woman in the second picture here and the woman in the third one, because here in the second one, you know, if you visit a friend's house and you didn't know that they have a dog and you're allergic to dogs, you know, that's kind of bad. I feel really bad for you, right? Even the dog feels bad, right? Or if you show up in your workplace or you know, cafeteria, and one of your coworkers or classmates has peanut butter, right? And you're very sensitive to peanut oils, right? I feel really bad for you also, right? I mean, these days, more and more, there are restrictions placed on, for example, kids, what they can pack in their lunches to bring to school with them. Right? I think when I was a little kid, I'm not even sure I knew anybody who had a peanut allergy, right? And we can bring anything we wanted to eat for lunch. Right? But these days, like my kids in grade school, we have this long list every semester of things that they're not allowed to pack in their home lunches, right? Including obvious things like peanut butter or other types of nuts, but even things now like strawberries. I think one of my daughter's classmates has an allergy to watermelon, you know, stuff like that, right? So again, more and more of these kids are becoming sensitive. You know, the guy in the first picture, I'm not very sympathetic, right? Because I think if you know that you're allergic to flower pollen, I don't know why you'd end up standing in a field of wildflowers like this, right? So as far as I'm concerned, this guy did this to himself. Right? Okay. When we talk about hypersensitivities, it turns out that all allergic reactions follow one of four different categories or one of four different mechanisms. You know, so that might be kind of surprising to you, right? That all allergies can be categorized as one of four types, and we just call them types 1 through 4. That's what these cartoons are illustrating here. And we'll go into them in a little bit more depth, but this first slide just introduces them. Types 1, 2, and 3 all involve antibodies. Right? Type 1 involves IgEs, and the way that IgEs are sometimes suited up on the surface of a mast cell. Right? Type 2 involves IgGs, IgGs that are recruiting the activity of things like macrophages, right, to attack cells that are presenting something. Type 3 involves immune molecules like antibodies improperly cross-reacting, causing sort of like antibody to antibody sensitivity that can lead to chronic problems. Type 4 does not involve antibodies at all. It involves T cells. Right? And we'll look at this in more detail. But the point is, now that you know, right, there are 4 types of hypersensitivities that explain all allergies. 3 out of the 4 types involve antibodies. The 4th type involves T cells, but all 4 of these involve components of our adaptive immune system. Right? So that's why we say that hypersensitivities arise from some mistake along the way in our adaptive immune response that gives rise to these types of situations. So let's start off with type 1. Type 1 has to do with the binding of IgEs to a particular antigen. So you'll recall that IgEs are these big, sturdy antibody isotypes that don't have much of a hinge region, but they have a big stem region. Right? Then we previously learned that IgEs are the most useful for fighting against things like large multicellular parasites. Right? So imagine, for example, that in a normal course of an immune response, maybe the body's infected with some sort of a parasite like a worm, something like that, right? There'd be plenty of antigen molecules that are being shed by that worm, right? So this molecule here, it represents the antigen, and it has certain types of little epitopes on it, right? You would have mast cells. Remember, these mast cells are these granulocytes that contain all kinds of inflammatory, sort of noxious molecules in them. And if they are triggered or stimulated, they would then release all of these types of molecules into the surrounding tissue. So these mast cells, even though they are innate effector cells normally, because they can be suited up with IgEs on their surface, because these mast cells have Fc epsilon receptor. So remember, Fc just means the stem part of an antibody. The epsilon means specifically the stem of IgE, right? So this green Fc epsilon receptor hangs on to IgEs. So the whole surface of this mast cell is actually equipped with different IgEs. It doesn't have to be the same type of IgE, because after all, these IgEs were just floating around, right? And then they just all got picked up sort of randomly by the mast cell. So in this case, in order for this mast cell to activate in response to this antigen that maybe comes from being shed from a worm or something like that, it needs two or more of these Fc epsilon receptors hanging onto IgEs to cross-link. You see how in this case, right, two IgE molecules bind, therefore two of these Fc epsilon receptors stimulate. And that is what then stimulates the release of things like, or the degranulation and release of things that are contained in all of the mast cells, vesicles inside. Right? This is important because this is a way to make sure that mast cells aren't, too sensitive to things, right? Because if all it took was one IgE to have one antigen bind to it, then this mast cell, I think, would be stimulated a little too easily, right? So this type of a mechanism, this type 1 hypersensitivity, is what's behind certain types of allergies like hay fever, right? Allergy to pollen, the allergies to animal fur, food allergies, bee sting allergies, right? These are all type 1 allergies that involve IgE. We call them immediate hypersensitivity because the reaction is very fast, right? Because if you're exposed to any of these types of antigens, then you better believe, right? If your mast cells have the right IgEs on them, you'll get a lot of this kind of IgE cross-linking and activation. Right? And because of that, remember, where are these mast cells located? The mast cells are not just floating around in your blood, they are situated in your solid tissues. Which tissues in particular? Mucosal layer tissues, right? Because that's where you actually worry the most about being exposed to something like a parasitic worm, right? Because if you eat some food that's contaminated and has, like these parasites in it, it's going to be the mucosal lining of your GI tract, right? That gets first visited by these worms, right? By the parasite. So therefore, the mucosal layer is loaded with mast cells that have various IgEs on them. And as a result, right, all throughout your mucosal layers, you have these mast cells hanging onto different IgEs. So if you get exposure, then the reaction to these mast cells releasing all these things like histamines or other types of very irritating molecules will affect something like your entire GI tract, upper GI tract, or respiratory tract, right, if this is where it's happening. And it can be very life-threatening. Again, because once this activates in the mucosa and you have all this type of crazy sort of inflammatory type of response being sent out by the mast cell. You might get constriction of smooth muscles that would then potentially limit, for example, your airway, or you may actually have the opposite result on the endothelial cells of your blood vessels. They may become very dilated, right, to try to increase things like blood flow. And that can have a dramatic effect on things like dramatically lowering your blood pressure all of a sudden. That's why things like bee sting or certain types of food allergies like nuts, right, are so life-threatening, right? Because if somebody who's allergic to those things either gets stung by a bee and the venom of the bee spreads through their blood and triggers all these mast cells, or if somebody's exposed to peanut oil, for example, and begins to line their throat as they swallow it, right, they can have very serious reactions. What does this imply? The fact that you have IgEs that are sensitive to detect these different types of antigens tells you that you were previously exposed, right, to this antigen. And that's why I think many of you know that there's this saying that if you are stung by a bee the first time in your life, you're not going to have any kind of allergic reaction, right? But if you're the type to develop allergy against bee sting, you will know it the second time you're stung by a bee later on. Why? Because the first time you're stung by a bee, you didn't have IgEs previously selected for to recognize bee venom, right? Because why would your body have ever known to select for IgEs for that purpose, right? But after you're stung the first time, a mistake is made. And for some reason, your adaptive immune response begins to select for B cells that have the right antigen binding domain to bind to, whatever the components are of the bee venom. And then over weeks or months, you develop immune memory, right? But in this case, it's mistaken immune memory, right, in the form of these high quality IgEs. So that the next time, right, you get stung by a bee, this happens. Type 2. Type 2 hypersensitivities also involve antibodies, but in this case, we're talking about IgGs. So in type 2, if you're exposed to certain type of compounds or certain types of medications, things like penicillin, for example, maybe a number of you in the course know that you are allergic to penicillin because of a bad experience that you had when you were a kid. And so now on your medical chart, right, it lists a penicillin allergy. This is why when you go to your doctor's visit, you always have to fill out a little thing to just remind them whether or not you're allergic to certain types of drugs. Right? So penicillin, of course, is an antibiotic. But the way penicillin works in the body is that a side effect is that when penicillin is inside the body, it can covalently bond to or bind with different types of molecules that are on the surface of your normal body cells. That's not the main mechanism by which it works, right? Because it's supposed to be targeting the things like the bacterial cells. But an unfortunate side effect is the compound itself, right, can interact and bind to covalently molecules that are on the normal surface of your normal surface, normal, happy, healthy body cells. So that's what we see here. These blue cells represent normal cells in your body, right? The penicillin is sort of this hot dog, right? And it comes and it binds to certain molecules on the surface of the cell. Well, these sort of look like corn dogs, right? But all right, forget that. That's just going to be a confusing analogy, right? So what happens is the shape of these molecules or these proteins on the surface of the normal cell gets altered, right, because of this. And as a result, it's quite possible that you have naive B cells that were sort of going to work, right? Chances are they were never going to bind anything that was ever any useful. Oh, but it turns out by chance, right, this naive B cell actually binds to this new weird looking sort of composite structure here. And therefore, this B cell might be improperly encouraged to be selected and to start the process of undergoing affinity maturation to make better and better versions of this antibody. Okay. So that means in the future, if you are treated with penicillin again, right? So keep in mind, again, the first time you're treated with penicillin, nothing bad happened, and the penicillin probably just worked very well to, rid you of your bacterial infection, right? But the second time, maybe months or years later, you already have these high-quality IgGs around as part of your immune memory. And as a result, when those high-quality IgGs see the combination hot dog, corn dog, I know I wasn't going to use that analogy, but I like it, right? Then it now goes to work, right? And as part of your secondary immune response, these IgGs begin to bind to your own body cells, and they start to attract or recruit the activity of macrophages, right? Because these IgGs start causing things like complement proteins to be attracted here. And these complement proteins make it very easy for a passing macrophage to grab on. And in this case, then the macrophage would attack and start damaging your own body's cells. Okay. Type 3 has to do with antibodies improperly cross-reacting. This is really interesting if you think about it. An antibody is a protein, right? It's a protein made of 2 light chains, 2 heavy chains. We certainly looked at the structure of antibodies quite a bit this semester. And the ends of the variable domains are unique shapes in order to bind to other types of 3-dimensional shapes, right, that represent an epitope that this particular antibody binds to. So because antibodies are just proteins, right, there's nothing to say that one antibody, might turn out to be by chance, crazy chance, the right shape to bind to something like the variable domain of another antibody, right? So in this case, you have yellow antibodies binding, or blue antibodies binding to, yellow immunoglobulins here, right? Now, this would pretty rarely happen inside of your body normally. Why? Because you think about it, right? In this scenario, they're talking that these blue antibodies came from the outside. So these were antibodies that might have been manufactured as mouse antibodies that are then purified and used as a pharmaceutical treatment, right? Infusing antibodies in to the body so that they can go ahead and detect certain types of signals is something that we already looked at, right, as ways of maybe preventing different types of immune responses. Right? Certainly, we talked about in the case of pregnancies, right? If an Rh negative mom, right, delivers an Rh positive baby, we learned that there's a very good chance that as a result of childbirth and the trauma of childbirth, that the mom will end up developing antibodies against the Rh positive epitope. Right? So we said that the way that physicians often deal with that is to infuse high quality antibodies into the mom during late pregnancy to kind of discourage her own naive B cells from being selected or chosen, right? And so that way she doesn't develop an immune response against the Rh positive blood type. So here's another example, right? These blue antibodies might have been infused into a patient for some therapy, but unfortunately, the patient has, by chance, a naive B cell here whose Ig, maybe this is IgM, right, is the right shape to bind this, and it may cause this B cell to be selected. And when this B cell is then selected, what will happen is that it will then start producing lots of higher and higher quality IgGs, the yellow ones. Right? And if this patient continuously needs to have infusion of these blue antibodies, what will happen is you start having antibodies attacking antibodies. And what happens there is you get big sort of immune complexes, sort of masses of antibodies binding together, and your body has a hard time dealing with that. Especially because you think about it, these clumps of antibodies then, it almost represents precipitations of sort of masses of protein, and that can cause things like the blockage of capillaries. So why I said that this is typically, not a problem if you're just talking about your own antibodies, right? Because why couldn't you have a situation where you make a blue antibody and a yellow antibody? They're both your own antibodies, but by chance, unfortunately, they detect each other. It's very unlikely that would happen. Why? Because even if you made, let's say, a B cell that makes the blue immunoglobulin and a B cell that makes the yellow one, right? What are the chances that in the body these two would ever interact with each other? Right? It's pretty slim. So normally these type 3 types of immune complex problems happen when you are constantly being exposed to a foreign antibody that your body then has plenty of time and exposure to build up an improper sort of immune response against. That brings us to type 4. Remember, there are 4 types of hypersensitivities. The first 3 types, 1, 2, 3, have to do with antibodies. Type 4 is mediated by T cells, not antibodies. Type 4 allergic reactions are what happen for those of you who have ever had allergic reactions to certain types of jewelry, right? So, those of you who wear earrings, I'm sure you've had situations where you wear a certain pair of earrings that you just got, and then after maybe a few days, right, you feel like your earlobe has gotten infected, right? It's red, it's tender to the touch, there may even be a little bleeding or oozing there, right? My daughter, she's very sensitive to different types of metals. And so, cheap sort of cosmetic jewelry, stuff like that, she's very sensitive to. Her earlobes turn red and she gets a little bit of bleeding, right? So as a result, she insists that, she can only wear real gold earrings, right? So, but anyhow, so why would that make a difference? Gold versus cheap other metals. So oftentimes, cheap cosmetic jewelry is made from, not gold, not platinum, not even silver, but maybe things like nickel, right? There might be amounts of nickel used to make the jewelry. Nickel ions, when they're shed from the metal, because of course, right, as long as that metal is sort of pressed up against your skin, right, the little nickel ions are falling off of it and they can get into your tissues. They're going to get absorbed by your skin. These nickel ions actually form adducts. In other words, they form chemical bonds with certain amino acids, for example, histidine. So histidine, which is one of the amino acids, of course, that's used to make various proteins in our body, now it has this nickel group stuck to it. So as a result, right, it's kind of a different shape than what our body is accustomed to seeing. So as these nickel and protein adducts, represented by the red dot stuck to the nickel, makes its way into a lymph node, right, it gets presented to different types of naive lymphocytes like T cells. Right? And so chances are you're going to have some T cells that, through their VDJ recombination, their shape of their T cell receptor recognizes these modified peptides that have funny histidines in them because there's nickel ions attached to them, right? They're a different shape. Right? And they'll activate. Right? So then you'll select for T cells that are both cytotoxic in nature and helper in nature. You can see they begin to multiply, and then they go through your lymph system, eventually get back to the blood, and they might get back to your earlobe. Right? And that's where they start seeing, whoa, right? Tons and tons of otherwise healthy epithelial cells here that are part of your earlobe, right, are showing this peptide because of all the nickel from the earring. So these T cells go to work, right? The cytotoxic T cells start killing your earlobe cells, right? Thinking that they are somehow infected with something bad here. Right? And also the helper cells start recruiting macrophages and other immune cells to similarly try to fight this this perceived threat, right? And that results in the tissue in your ear being damaged. So, I was talking about how my daughter, she insists that, Dad, I can only wear real gold. And I'm like, all right, whatever, right? But actually, it's true, right? Because I think all of you know, those of you who do wear earrings, right, that gold does not form, well, you wouldn't know this, right? But gold does not cause similar types of allergies. Why? Because the gold ions that fall off of your gold jewelry, they do not form these types of covalent adducts with amino acids like nickel does, right? Okay. All right. So that is sort of an overview of these four different hypersensitivity types. All right. So we'll spend some more time the rest of this lecture and next looking at some examples. So let's start off by looking at type 1 again, right? Because again, I think this one in many ways is kind of the most exciting one because it's the one that typically when you think of somebody with allergies, especially deathly serious allergies like shellfish, peanut oil, or bee stings, right? It's because of this. So remember, IgEs are normally useful for defending against multicellular parasites because a multicellular parasite is way bigger than something like, an individual immune cell, like a macrophage or a neutrophil, right? And for that matter, a huge, massive multicellular parasite like a worm, it doesn't care about things like complement proteins trying to attach to it, right? It just kind of laughs it off, right? It's kind of silly, right? So you have IgEs because IgEs are much stronger, so they can bind to the parasite and importantly allow what, right? They allow things like mast cells to then be brought to the parasite. And if you have a whole bunch of mast cells, then degranulating, right, that releases a whole bunch of compounds and chemicals that can stimulate oftentimes very violent physiological responses in your tissue in an attempt to get the parasite expelled. I want you to consider something, right? A parasitic worm, a multicellular parasitic worm, even though you might want to not want to sort of accept this, biologically, a worm is a lot more similar to the cells of our own body, the eukaryotic cells of our body, than something like a bacterial cell or a viral particle or a fungus cell, right? So therefore, parasitic worms, right, if we have to mount a defense against them, it's a little bit trickier, right? Because the cells of a parasitic worm might not really be all that different than the types of eukaryotic cells that make up our body. That's why when we're fighting against worms, for example, these are a couple of helminthy worms here, the type of T cells, helper T cell, that's preferentially put to the job are these Th2 T cells, helper T cells, right? CD4 positive T cells. The types of cytokines that are used are seen here, right? And the specific types of antibodies that play a role in this are IgEs and IgGs, but not just any old IgGs, specifically subtypes of IgGs, IgG1 and IgG4. All right, and we'll take a closer look at what the big deal is there. Right. So the cells that make use of the IgEs to try to fight against the parasite are things like eosinophils, basophils, mast cells. These are your classic kind of granulocytes. Right? So when these cells blow up and release things like histamines and other types of contents of their vesicles, right, this leads to your tissue being irritated. And so that's why you sneeze or you cough, or maybe you even vomit if it's a particularly strong reaction taking place inside of your stomach, or you get diarrhea, right? These are all things, right, that are associated with type 1 allergies, whether from breathing something in, right, or eating something, right, like food poisoning, for example, right? And it's an attempt of our body to try to expel different types of parasites, right? And all of it's a result of severe inflammatory actions by granulocytes degranulating. So what about these different IgG types that I mentioned? For most of this class, we've just talked about IgG as just one of the 5 types or isotypes of immunoglobulin, right? You have IgM, you have D, you have A, you have E, and you have G. So now what I'm showing you is that within the IgG group, you have slight variations of IgG, 1, 2, 3, and 4. That's what these cartoons show here. I mean, each one still kind of fits the description of an IgG, right? It has a very modest-sized Fc region, the stem part, and it has a hinge region. Right? Remember the hinge region, the little bars that go across in black here represent disulfide bonds between cysteine amino acid residues that are on the opposite sides of these chains here, right? And they form these disulfide bonds that are pretty strong, right? And they form this flexible hinge that allows the two arms of the IgG to kind of flip around a lot, depending on what they're binding to. So the IgG 2, 3, and 4 are just variations on that, depending upon how extensive and flexible the hinge region is. Right? So we're not going to talk a lot about IG2 and IG3, but we will talk a bit about IG4. Right? So these different subclasses of IgGs, they work together with IgEs as part of a typical type 1 hypersensitivity response against something like a parasite. Right? I want you to remember that the ability to class switch, in other words, a given B cell that starts off as always making IgM, but then is told by helper T cells to class switch into making IgA, E, or G, it does so by holding on to the same VDJ rearrangement that it came up with, right? Because after all, the VDJ recombination is what determines that it has the particular antibody antigen specific shape, right? So you don't want to lose that. But what you want to swap out are the stem regions, right? That make it a different antibody isotype. So here's mu, right? So IgM would involve using the mu block as the stem. Here's delta, so IgDs would be those. And then here you see, here are the gammas, right? So there are different blocks for gamma 1, 2, 3, and 4. And also here's the alpha, alpha 1, alpha 2 to make IgAs. And here's the epsilon block, right, to make IgE. So as an infection with a parasite proceeds, B cells will be undergoing affinity maturation through hypermutation, and they'll be switching through these different types of Ig isotypes. Right? When it first starts off, you might imagine that there might be quite a bit of inflammation going on, right? As you originally have B cells that are stimulated and you're sort of mounting this defense. However, in the case of things like parasitic infections, we actually don't want a lot of inflammation, right? Because inflammation, if you have a parasitic infection, might actually make it easier for that parasite to be able to gain access to what it wants, right? As it's making its way through your tissues, as it's trying to get a lot of nutrients, right? So over time, as the infection proceeds, the different class switching reflects a shift away from inflammation at first, more towards specificity. So this table here shows that during the course of an infection, as B cells are class switching between IgE, they may be told to change to IgG3 first, then IG1, then IG2, and eventually IgG4. On the right, what they're doing is they're asking you to kind of imagine that the number of different somatic mutations that are appearing in the VDJ locus, right, as part of hypermutation will be building up. Right? So as you get closer and closer to IgG4 at the end, right, you have more examples of mutations compared to what you first started with. Right? And in many ways, that's how they've determined that this is the typical progression of class switching. Importantly, IgG4s, right, function by discouraging inflammation from happening. IgG4 is a little weird, right? Because we've discussed before that one of the hallmarks of an antibody or an immunoglobulin, the Y-shaped molecule, is that both arms are identical, right? When it's being made by a B cell, right? A B cell makes one type of short or light chain, one type of heavy chain, right? And then an Ig molecule is two of those heavy chains, two of those light chains. So as a result, the arms are typically identical, right? And for most IgG types, it remains that way. But IgG4 is a bit different. Even though it's still made with both arms identical from the B cell, once different IgG4s are circulating around in your blood, if they bump into each other, they can actually swap arms. So in the blood, imagine this red IgG4 bumps into this blue IgG4, right? And they can actually switch arms. So you end up with an IgG4 that is not monovalent in terms of picking up only one type of antigen, but it's bivalent, right? It can pick up two different types of antigens. And why is this useful? Because this gives you extra flexibility in being able to cross-link or bind to different things that are all coming from, let's say, the same parasite, right? To be able to really heighten its detection. The other thing to keep in mind is IgG4, when something binds to it, right? Instead of encouraging the macrophage to release cytokines and inflammatory responses, it actually discourages the macrophage from producing these types of inflammatory signals. So here you can see, here you have, if it were a straight up IgG1, this is a normal IgG1, so both arms are exactly the same, and it's bound by the Fc gamma receptor by the macrophage. If something bound to it, that would stimulate the macrophage to release inflammatory cytokines. Right? But if IgG4s bind, they're held on by the macrophage, and they actually cause an interference. They stop the macrophage from releasing inflammatory signals. Again, why? Because when fighting something like a parasite, you don't necessarily want to have a lot of inflammation, right? So if the infection has gotten to the point where your B cells are making high quality IgG4s, right, as part of fighting the parasitic infection, part of how those IgG4s are going to work is to tell the immune system, all right, let's cool it with the inflammation. Let's just focus on things like allowing the IgEs and the mast cells and the basophils to release their types of noxious compounds from their granules directly, sort of to make the parasite itself sort of uncomfortable, right, or irritated or disrupted, right? So IgEs, we said, are very sturdy and they're held onto on the surfaces of mast cells. You can see that IgEs, you don't find them in the blood. They're preferentially found in the tissues because that's where they're going to do their job, right, especially in the mucosal layers. The way that they're held onto by the Fc receptors of mast cells is kind of funny because in the cartoon over here, this is the super simplified version to just make you think about the fact that you have Fc epsilon receptors, the green, that are holding onto the Fc regions of these IgEs. But the reality is this cartoon on the left shows how they're held on, right? So this Fc epsilon receptor here actually grabs on, to sort of like this part of the stem, really close to where the two arms come off. And as a result, these two, the Y-shaped antibodies kind of crumpled up, right, and folded. That's so that it can have a really good grip and hold on to this IgE very tightly. So look at this. Think about this, right? B cells that are producing a particular IgE, that maybe there was a mistake made and the B cell thought, I need to make this IgE because, my antigen binding domains on my immunoglobulins are recognizing this foreign antigen, and for some reason I've been selected like this is a threat, right? A given B cell, right, will only have on its surface that one type of immunoglobulin, right? So for example, if this yellow immunoglobulin detects an antigen on a parasitic worm, like a herring worm, right, the whole surface of this B cell only has this on it. Or if that B cell, so that would be a good situation, right? Because this is how you have adaptive memory that's needed to protect you from future infections by this true parasite, right? But the same thing applies even if there was a mistake, right? So in this case, this B cell made immunoglobulins against pollen, right, from flowers. And hey, it was told to be selected, right? But the same thing holds true. Its surface only contains immunoglobulins against the pollen, right? Nothing else. However, think about the mast cells that make use of these antibodies that are floating around. Because they just have these green Fc epsilon receptors, they can just grab onto any IgEs that are coming by, right? No matter what that IgE type is. That means you can have a mast cell that's very sensitive to a herring worm or a blood fluke worm, a hookworm, or a roundworm, right? And that's good because this mast cell, boy, this is a valuable mast cell, right? It protects you against a number of different parasites. Oh, but the problem is, what if it's the situation where the IgEs that are being made are actually mistaken IgEs, things like pollen or cat dander or peanuts or bee venom, right? Now the opposite is true. This mast cell is making you very hypersensitive to various types of allergies. Okay. This is the last slide for today, and we'll just stop here. Like we mentioned before, immunotherapies might involve actually intentionally infusing different types of high quality, high amounts of purified antibody into a patient with the intent of those therapeutic antibodies binding to things in a way that's desirable. Right? Oftentimes, it's used to interfere with certain types of things. For example, there are therapeutic anti-IgEs that are produced to help people with severe allergies. So in this case, let's say the yellow IgE is the one that the person's atopic system is making mistakenly, right? So for example, maybe this is an IgE against things like pollen or dust or cat hair, cat dander, right? As a result, if the mast cells were allowed to grab onto these IgEs, right, then the mast cell would become sensitized to that particular allergen. In some cases, that might be very debilitating and get in the way for that person to be able to live his or her normal life. Right? So one therapy might be to infuse into their system these brown antibodies, these high quality antibodies that are designed to bind to the stem region of IgEs. Right? So as a result, they prevent the IgEs from being picked up on the surface of the mast cell. So that would be good. So there are examples of therapeutic antibodies to treat allergy sufferers, right? Severe allergy sufferers. But at the same time, there are also cautionary tales. There are examples throughout history of certain types of therapeutic antibodies like this, right, taking a bad turn. Right? So in this case, what has happened is that the therapeutic antibody, maybe it was an experimental one, right? So during clinical trials to see if it's working in people, this might have happened. Instead of binding to the stem region of the IgEs like they were designed to do, look at what happens here, right? They've actually ended up binding to or being detected by the antigen binding domains of the IgE. And as a result, they are overstimulating, right, mast cell activation. And in this case, it could be really bad. And so in a couple of cases of clinical trials, I think in the early 1990s, right, some people actually got really, really sick during clinical trials using these antibodies because of this problem. Okay. All right, so we'll stop there today. There's one more slide, but we can start next time with that slide. All right. Stop the recording.